Well, right, thank you. Thanks for the invitation. And I thought I saw where this is somewhat informal. Questions should be thrown at me as I'm going. So one of the first things I started thinking about when I moved to the great city of New York, into an academic arena, a big medical center, was sort of the types of rules and beliefs that were held within the system, and whether these beliefs and rules were any longer true or optimal, or whether we should hold to these. A very conservative, paternalistic environment. So started thinking about orthodoxies, and is it time for us to start questioning what's happening in medicine, given the types of biases that get introduced into this sort of industry as well as other industries. And we see lots of evidence of this throughout human history. And one of my favorites is on the Warner in 1927 proclaiming who's ever going to want to hear an actor talk in a movie, or there'll only be a world market for 5 computers. Sort of whatever the narrowness of the thinking we get locked into, these are the sort of the beliefs that get solidified, and at some point we have to start questioning those and be disruptive. And so my thoughts have been coming to Mount Sinai, which is now the 4th largest medical center in the United States, 4 million patient visits a year. It's a pretty big complex, but is this sort of fundamentally poised for transformation? And one of the things we see that's driving this is, of course, this this big data, our ability to both generate big data and apply sophisticated algorithms on high performance computing to learn things from such data and then drive decision makings from that. And we see this across many different disciplines. So if you look at the Oracle team for the America's Cup, they have these pretty sophisticated boats where they now have 300 plus sensors placed throughout all aspects, monitoring all aspects of the performance of this boat and the crew. And they're acquiring data at around 10 hertz, so thousands of variables being monitored through these sensors. They're generating around 300 gigabytes of data a day, and of course crunching on that data to sort of tune and optimize the performance of that boat. And in fact, at the end of the day, they're dumping the servers off the boat into the water, and another boat comes and picks them up, and then they're doing more high performance computing on it overnight to try to, again, better tune the performance. We see the same sort of thing in the Indy cars. Team Honda, again, has very sophisticated number of sensors looking at all aspects, engine, clutch, gearbox, and so on, and the driver, the health of the driver. And again, so acquiring around a gigabyte of data each lap in real time, adjusting the performance of the machine and the driver to, increase their chance of performing better. But contrast that to the human system, which we should maybe care a little bit more about. And what we have in the great state of New York, when you're born, a newborn, is they'll take a little prick of blood from your heel and smear it on a piece of paper, and they'll mail it up to Albany, and then they'll run a little test on there to score maybe 40 or so variables they'll score, and they're important because they're going to tell if there's something really catastrophic going on in the newborn, but a pretty far cry from what Team Honda does and Team Oracle does as far as monitoring the performance. And of course that happens the first day you're born, and then you're out and you're on your own. So we're seeing a revolution though, and can we perform the same kinds of monitoring technologies on humans that we perform on machines? And of course, no shortage of companies now popping up with all these physiological censoring devices that you can buy now in your neighborhood stores. One of my favorites is coming from Scanadu, which has a handheld device that you're like waving over the body, and it's making like 10 physiological measures, connects up to your iPhone, and sends that away for interpretation if something's going on, and can engage physicians. So this group is in the lead for the Tricorder X Prize for such a device that you might have seen on Star Trek in the early days. We have now printable tattoo biosensors that again can pick up fairly sophisticated physiological measuring of what's going on in your system. These are Bluetooth enabled, so we can connect up to your iPhone and dump data as it's being collected. And not even the babies can escape the grasp of this technology with Pixie Scientific. now embedding sensors in the diapers to do some censoring of what's being excreted from that baby, and so I think cool. And then companies like Theranos popping up now in your neighborhood Walgreens, where with a very small amount of blood, you can simply go in to that station and have a very, very wide array of tests run, right on the spot, right at Walgreens. So again, the closeness of all the medical advances coming, into your neighborhoods for ease of testing. And I can't keep the NSA out of this big data collection effort on what they're learning about us, in all aspects of our lives. And maybe someday we can convince them that if they open that data up, we can actually do some good for humankind on the wellness front. But sort of a lot that we're able to learn from that scale of operation that they're putting up there. And even in the state of New York, if you go to the NYU Center for Urban Science and Progress, they have this cool, I forget actually now the name of it, but it's basically leveraging the terabytes of data that New York is generating from all the traffic flow to electricity usage and very localized information for those sorts of things. And this kind of data now becoming available and able to access it and start integrating that kind of information with maybe more molecular information on the cell. And of course, what we want to do with all of this is aggregate it, compute on it, and then present real-time decision guidance in the form of different apps running on your smartphone or on the web or whatever. So the whole vision that we have then in this whole data universe and how it can be disruptive in the medical arena is one of aggregating all of this information, not just the molecular information on you, but everything that's happening in the context of yourself, building predictive models, and then using these predictive models to guide decision-making regarding diagnosis and therapy strategies for an individual. So as an individual walks into the Center at Mount Sinai, the idea would be leverage the digital universe of information, point that at the individual, take their own information in the context of the digital universe of information via these models, and just simply better diagnose and figure out better treatment strategies for those individuals. So we're pursuing this at Mount Sinai. At present, we have a biobank of 30,000 individuals where we're piloting a lot of this, where many different dimensions of data are being collected on these individuals. They're all consented for recontacting. They've all been exome typed, and we're, of course, have access to, since they're identified for recontacting, we can hook their information up to the electronic medical record system, and any test that's being generated on these individuals, any study they're participating in, we can aggregate all of that data, and have begun piloting studies where these individuals are putting on the wearable devices to collect other high-dimensional data on those individuals to hook up to the molecular data. And the main aim is one of connecting all of that molecular and other such data to the physiology of the system. So we want to create different types of models, and we view these as networks, but we want to hook that up to the physiology of the system so that we can better predict when that physiology is going awry, and if you're in a pathophysiological state, how to turn that around to make you more healthy. But how you view this information matters, and so if you read some of the behavioral science, you'll read things like this that I thought well reflected how individuals are presented with information can have a very big influence on your ability to solve or innovate. And so this was a study that was carried out where two groups were presented with the identical information, but how they were presented with it was slightly different. So the problem was to affix the candle to the wall and light it without wax dripping on the wall or the table. So that was the problem to solve. They were both presented with this information, but in group one, they were presented with tacks inside the box, and group two, tacks outside of the box. Other than that, it is completely identical. And pretty amazingly, the group two that was presented with a tacks outside of the box came up with a solution twice as fast as individuals in group one. So again, just that simple perception problem in this very simple setting, sort of illustrating that how you perceive that information can have a big effect. And so my thought is that the more expert we become, the deeper these grooves become in our brain, the more narrow we become, and the less likely we are to innovate or create, because we become sort of blinded in the field, and we're trained by the same people, and we're sort of this inbreeding that goes on, and it ultimately weakens your ability to innovate. And companies have, this is not constrained to medicine. Lots of companies have this problem, and they've thought of ways to break out of that. Like, how do you break out of that type of narrow mindset? And so here are a few examples that I liked when I heard about it. One of them, my favorite, is BMW. So I'm just kind of a little bit of a speed freak and like to drive an S1000R super bike and an M5, and so was really curious to learn that when BMW wanted to, they want to make their cars better and better performance, so they sat engineers with the owners of the cars to drive around and to learn, what are the owners really after? Do they want, better acceleration or better cornering or better braking? And so when they came back, they were shocked at what they learned, which was that the number one concern of the owners of BMWs was preventing damage from pulling into or backing out of a parking spot. So they, it wasn't anything to do with performance. It was that they spent a lot of money for this car. When they backed out or pulled in, they were dinging it. And so that's when they innovated the park assist features. So BMW was the first company to pioneer the use of park assist features, and it was driven not by anything they thought as experts about performance of the car, but it was by actually going to the customers. And another one I like is Bowel.
You need expert by definition of the one who studies, right? So that's the definition of expert, the one who doesn't look at what he does, right?
Yeah, it's a good, I like it. Apple's another good one. When Steve Jobs went to Wall Street and said, I'm going to make stores where you go in and you buy electronics, he was completely laughed at. There was not an analyst on Wall Street that said that was a good idea. They all said Dell had changed that model forever, and what people do is they buy electronics online. They don't go into stores and buy electronics, but certainly we know how that ended, and a very successful endeavor that he launched there. So again, not a foreign idea to break out of your narrow mindset and to innovate, but you have to approach it in the right way. So the way we think about this is one of measuring lots of different dimensions. So we're now able to measure many different dimensions of a system longitudinally, and we can now integrate this information in ways that build predictive models of those systems. So that's sort of the approach we're taking to break out of our narrow mindset, and that's when we look at how DNA and proteins and metabolites and RNA are hooked up. They're not hooked up in simple linearly ordered pathways. They're hooked up in very nonlinear networks that define biological processes, and these networks don't act in isolation. They act in concert in a network of networks. And once we start modeling in this very data-driven way, where we don't accept any known pathways a priori, we're saying let the data speak, and we'll derive that knowledge from the data. We can start looking at how things at a network level, like how do changes in one part of the network affect other parts of the network in the system under study. So this could be a single cell or tissue type, but of course we're interested in the entire system. So what we really want to know is not only what's happening within a given cell or tissue, but as we make changes in one part of a system or tissue type, how does that propagate through to other parts of the system to make effects on other tissues or other parts of the physiology of the system. And once we have this sort of model, if it's accurate enough, we can start carrying out biology on that model, right? We can start doing perturbations on this model in silico on the computer, not having to do the studies in humans and or other model systems, and we can see how the perturbations affect the system. So we may perturb some part of the system over here, and we can model how it's rippling through the system, how the system equilibrates, and in that equilibrated state, are you in a more favorable or less favorable condition than you started? So those are the kinds of games we can play. Of course, to do this kind of work, we need access to pretty high performance computing. That's something we've invested pretty heavily in at the Icahn Institute, and have hired guys like Jeff Hammerbacher, who you may not know, but he's now built a couple of supercomputers for us. So Jeff was the founding chief data scientist at Facebook, so he's classmates with Zuckerberg, and was the guy who scaled Facebook out of Harvard to the big time. So the guy who, how do you handle exabyte scales of data and high performance parallel computing and a Hadoop framework. So he's a guy who pioneered that. He then left and started Cloudera. which is how to bring Hadoop-style computing to the masses. They just closed a $900 million F-series round of financing, a $4.3 billion valuation in a 2-year period of time, pretty amazing. But have this sort of resource now at Mount Sinai to play this sort of big data, high performance computing game to build these models. And one of the keys of what we're doing is how do we causally infer, make causal inferences between variables that we're monitoring, given we're monitoring so many variables. So here's 2 variables we're sampling from a population, and if you look at the values of these 2 variables as we're sampling through the population, we may be looking at all the data in, say, our biobank, and we see that these 2 things are clearly correlated. And so this correlation-based information is interesting, but it's not going to tell us pairwise at a time what's causing what, or whether they're causally related or not. When you start looking at hundreds of thousands of these variables at once, you'll get interesting patterns of correlation. But again, how do you infer causality from that? And the way you classically do it in biology is then you may go observe a correlation in the population, and now you want to study, you have a hypothesis that, okay, this trait is causing this trait to change. That's your hypothesis. You set it up in an experimental system, and you're going to basically go and set up the system. You're going to make a perturbation to that one gene, and you're going to see whether you see a response in the other gene or clinical trait. And if you see that sort of cause-effect relationship, then you make causal inference. So that's the game in biology, but of course the problem with the game in biology are these perturbations are artificial. So knocking a gene completely out or overexpressing it at 10,000 times its normal amounts is a very artificial perturbation to the system, and the response to that system may not be appropriate from the standpoint of how it should equilibrate to operate normally, and so you could get adverse effects if you were to do that in a human, and then of course it's limited.
How do 10,000 times? How we can express so much? 10,000.
How?
10,000 times overexpressed. How we do 10,000 overexpressed?
So you put you engineer a construct where
it's 10,000 genes.
Well, not 10,000 genes, but you put the gene that you want to overexpress into a viral vector, and you put a promoter on it that basically amplifies the ability of that gene to be expressed at really high levels. So you're basically, maybe in nature you only see this gene at 1,000 to 10,000 copies per cell, but you can make a construct where you can make it 100,000 or 1,000,000 times, which is then very unnatural. Like the system didn't evolve to accommodate that sort of thing. So one of the things we had pioneered around 10 years ago, actually more than 10 years, was one of leveraging nature's perturbation source, DNA variation, as our systematic source of perturbation to infer causality. So this whole idea of Mendelian randomization was something we had actually first published on in 2003, where the idea was changes in DNA, that we can track that lead to changes in molecular features that in turn lead to higher order changes in physiological features. That sort of genetics game, we could leverage the Mendelian randomization through meiotic division as our sort of randomization source to make a causal inference. So think about it as a clinical trial, right? When you run a clinical trial, how do you establish causality in the trial? Well, you set up a randomized control design. You have a placebo arm, you have a treatment arm, and the key to inferring the causality is randomization of individuals entering the study to each of the arms. And so that's the key. And then when you see the subsequent effect in the treatment arm over the placebo arm, then you can make the inference that the result of that is a consequence of the drug. So in nature, what we can do is we can say that a given variation in DNA, so here is what we'll start with. We have traits, genotypes, and another trait, and they're correlated. These 2 traits are correlated, but they're also correlated with the genotype. Oops. Sorry about that, I have to use a keyboard to control this. So we can stratify a given genotype locus into, or given locus into the different genotypes that are represented in the population. So there'll be 3, at a typical SNP marker, there'll be 3 genotypes represented. And so what we're looking at are the correlations between people who carry this genotype and the traits that we're observing, and people who carry this other genotype and the values of the traits they're exploring. So is the pattern of changes in genotype correlating with changes in the expression or activity of a gene or a clinical feature? And given this correlation pattern, there's only one of three ways that these traits can be related. So again, the starting principle is change in DNA correlating with two phenotypes, and we can make the causal inference because those genotypes in the population are assigned randomly to individuals. That's Mendelian, the random assortment of chromosomes ensures that we're going to have a randomization pattern where this causal inference will work. And so given the starting position, we know that either phenotype 1 is responding to the DNA variant and then subsequently phenotype 2, or vice versa, or the DNA variant is independently affecting these traits. So we can mathematically model each of the different conditions and then simply pick the model that's best represented by the relationship that's best supported by the data. So just to look at that graphically, hopefully this plays good. So again, the way this works is we have many, many variables, not just pairs of variables, but many different variables. We're using DNA variation as the perturbation source, a little bit of mathematics, and then we're using the data and the mathematics to make a causal inference between these two nodes. We can do that systematically over the entire variable space, and what we're going to get is a result is a network, a probabilistic causal network model, where each of the edges gives you not only that these things are correlated, but it gives you the direction, the flow of information, the causal.
Their expression levels are correlated. What's correlated?
Yeah, so it depends on what you're entering into the system. So it could be gene expression, it could be protein levels, it could be metabolite levels, it could be biochemical, insulin levels, like whatever you're entering in. Yeah.
Do you prefer that we ask questions at the end or ask questions during the talk?
I don't know how it normally works. I'm totally comfortable with.
You're okay with questions during? Okay.
Sure.
So I'm just trying to understand the extrapolation you did from that SNP correlation model to the whole networking process. The three genotypes of a SNP, AA, heterozygous AB or homozygous BB, associated with a particular phenotype is just a simple correlation analysis. Mm-hmm. But then you went from there to say, well, there's this whole networking effect. Can you just be a little bit more explicit what the leap of faith is?
Right. So the leap of faith is that that sort of test is now being carried out in a Bayesian network context. And so the way to think about it is we're employing Bayesian network reconstruction methodology, but now have as the mutual information measure that sort of searches through the space and tries to understand the optimal connectivity pattern of the variables. That mutual exclusion measure is now based on the QTL, the genotype information. So we're looking at the mutual, the conditional dependency basically of these variables conditioning on the genotypic information, which is our perturbogen.
Is the network assuming that it's a multifactorial phenotype? In other words, you're looking for contribution from multiple different genes, or is the network on the phenotype side that you've got many different phenotypes that may or may not be associated with a single gene?
Yeah, so the network model is not making any assumptions on what the number of loci that are driving the trait, whether it's genetic or environmental. All the modeling is doing is saying, if I have this space of variables and I want to consider how they're connected, I'm going to look at all triplets at a time, and I'm going to apply a conditional dependency model to say, how are these things related? What's the sort of structure of those three variables? And then the sort of the Bayesian network idea is you sort of do that over the entire space. You're assuming this this Markovian structure. So think of it as a very complex joint probability distribution, right? That's what you start with, where you have the joint distribution is based on, say, 100,000 variables, however many you're looking at. And so how would you get a handle on that big function without making assumptions? Well, you wouldn't. It would be intractable. So we make the assumptions in the Bayesian network follow sort of Markovian principles that say we can basically decompose this very complex function into a simple product of functions, where each function is sort of a given node. Its state is only dependent on the state of its parent. So that's sort of the assumptions that get applied. So not entirely accurate, but maybe accurate enough to derive some
Okay, are you going to give an example of this, or
Yes.
we just? Okay, great.
For sure.
In an
How
example, I'll understand what the network of variables is better.
How about the thickness of the lines?
Yeah, so the thickness of the line, so how you depict this graph, ultimately there are many different ways that you can choose to depict the visualization of the graph, and I'll go through some ways we do that. For example, the thickness may depend on the strength of the relationship, the confidence you have that this call is correct, those sorts of things. You could have multiple edges for any given edge. So some of our graphs, you can click on an edge and it will explode out into multiple edges that define what are the lines of evidence that are supporting this sort of relationship. So gene expression may be one, protein, DNA protein binding may be another, protein-protein interaction may be another, and so on. So I'll just show you, once we have this sort of structure, so we can do biology in silico, we can perturb through feed-forward simulation any of the nodes in the network and see what are those nodes, what's the predicted change. And in this case, we see when you hit this node, it affects the entire state of the network. So we call that a key driver node, which are ones we're sort of really interested in. Like, what are the nodes that basically regulate the state of the network, right? So there are lots of nodes in the network, and we can go through in silico and perturb every single one of them and say, what's the prediction for the response of that perturbation? We can do that multifactorially too. We don't need to do it one at a time. We can do it in pairs, triplets. And then you're looking at what's the response of that perturbation. And those genes that are altering the state of the network we're very interested in from a therapeutic standpoint or a biomarker standpoint. And I'll show you again examples of how that plays out in practice. So that was leveraging DNA variation as a perturbation source. We can also use time series information as a perturbation source. So we employ both Granger causality type ideas and work that into the Bayesian network reconstruction paradigm, but also just directly using dynamic Bayesian networks as another modeling paradigm. So time series information can also be used, and we have a paper coming where we've now integrated time series based information with genetic information to sort of unify that space. One of the where this all leads in the future is I thought very well captured by this young man, Jonathan Carr, a guy I just hired recently into my group. So he had a paper in Cell about a year ago that was the first ever fully computational model for a living system. So he completely modeled every aspect of the system, from the metabolic flux, so what's coming in from the environment, and all the biochemical pathways, the DNA transcription, the translation of the RNA, the expression of the proteins, the modification of those proteins, every aspect of the system. So this was a pretty simple system, single cell. So I forget the name of the bacteria, but it had around 500 or 600 genes, so it was one of the smallest bacteria.
It's a manageable bacteria.
What's that?
It doesn't exist. That bacteria doesn't exist. 500 genes, 5,000.
500.
500 don't exist.
Yeah. Well,
They don't exist, right?
well, I can look it up and yeah, it's like 500 to 600 genes, and I think the people who are infected with it are.
They have more
will tell you it doesn't
more
exist.
metabolism, right?
But yeah, I can give you the actual name. I am just blanking on the name. And it may be slightly more than that, but the bottom line is it's one of the smallest living bacterial organisms on the planet. And so again, just think, he modeled every aspect, completely computational. And now what you can do is you can start that model. You can say, given some set of starting conditions, launch the cell, and then the model's going and modeling all aspects of the cell and giving what metabolites are fluxing in will drive different processes, and you can completely model all aspects of that and uncover completely new biology, as he demonstrated in his cell paper. So here you have different cells that have been launched with different starting conditions. He's looking at growth rates of the cell over time. So here you're seeing the different cells that he's modeling computationally growing. You're looking at all the different, the metabolic, the DNA replication, the protein expression, all of these processes going on, he's modeling, and this is from the model. is the growth of this cell and actually the cell division. So he was modeling, looking at the phenotype cell cycle in this particular case.
Mycoplasma genitalium.
There you go. Thank you. And how many genes does it have? You don't know. So all of the changes you're seeing in this cell are completely driven by all the computational modeling that he's doing to see what the cell is doing, and ultimately it will divide. And the cool thing about this was they were then able to go in and do perturbations to the system, like say, well, what happens if I pull this gene out, or if I amplify this gene? How does that affect the cell cycle? And they did that systematically throughout the gene space, and uncovered completely novel elements of cell cycle that nobody knew about, and then, of course, experimentally validated it. So, very cool.
But how to spread away with this parasitic bacteria? It's very hard to make in culture. The parasitic bacteria, it's very hard to make in culture, make experiments with them. You can grow them independently. It's exactly the way to special bacteria.
Yeah.
If you can cultivate independently, they don't live on a dish.
Yeah, so I'm not sure. I'm not sure what their model. They may have had an in vivo model for growing this. I'm not sure what it was, but I think the bottom line is a representation of the future, something that should breed some amount of fear into any biologist that this day is coming. Although I think it's inspirational too, that you'll be perhaps better guided in your experiments by what you can learn in silico. So the way
525 genes.
5. That was close. So the way we think, so we're on that trajectory of wanting to get to that. We're not going to be quite there yet in the near term for human, but we are sort of in the midst of this transformation of, whereas yesterday we were thinking about things a gene at a time, a protein at a time, a phenotype at a time, to today having these amazing technologies that can look at entire genomes and transcriptomes and proteomes and so on. And tomorrow, of course, it's all about how do you sort of synthesize, unify all this information, integrate it, and build, derive knowledge and understanding from it. So I'll give you an example of how we've done this in a few different areas and where we're going in the psychiatric arena too, so try to hopefully better connect to this particular audience. But I'll start with inflammatory bowel disease, because this was one of the first places where we showed it on a pretty grand scale of how we could basically organize all the data that are being generated from the big GWAS, and IBD is maybe a poster child for amazing success in the GWAS arena. When we did this, I got involved in this study in 2012. They had 163 loci through the GWAS that have been identified. Those are highly reproducible, genome-wide significant results. And of course, they were seeing pretty strong pathway enrichments in these, and they were pretty excited about it, but they had kind of had no experience in IBD, but they kind of called me out of the blue and said, hey, we've got 163 genes from this, and we don't really know what to do. In some cases, we don't really know what the gene is. Even when we know the gene, we don't know, is the genetics telling us that if you knock it down, you're susceptible, or you activate it, you're susceptible. We don't know what pathways they're operating in, what the larger context is, what the right cells are, and so on. So a lot of information they didn't know, and so they were asking for help. And so what I did was I said, okay, well, let me see if I can interpret these 163 genes in the context of a network. So we're building all these different networks across lots of different...
This is a parasite of the bacterium. Whose gene is
Yeah, so these, at this point, are human. So
human
these are
genes.
163 human genes.
But you cannot knock them out. How you can knock out?
Right, you can. But we know from the genetics, by looking at the different mutations in those 163 genes that are basically tracking with inflammatory bowel disease, you could say that this genetic locus is causal, causing increased susceptibility to this disease or protection against it. So we have those 163 loci where we know this.
Are these mutations that destroy the protein, or do they?
Right, so I should say, so these are like maybe inappropriate to call them mutations, because these are actually common variants that are being associated with the disease. And so most of these are not occurring in the coding sequence of the gene. They're occurring in intergenic regions or in the introns. And so that was the whole problem, is you couldn't look at the variant that was associated with the disease. You couldn't infer from that what that variant was doing. Like, it wasn't as simple as, oh, it changed this protein, this codon to a stop codon, so now you have a truncation and you're losing that protein. Like, none of them are that easy. So it's very subtle. You maybe activated the gene and made it expressed at 2 or 3 times its normal amount, or you knocked it down to subtle perturbations that are accumulating and basically causing the disease. And so that was the complexity that they wanted to try to get their heads around.
It's one microbe or it is or it's the whole environment. It's one bacterium involved into that disease.
Definitely, in IBD, there is a very big microbiome component, for sure.
No, this particular bacterium or the whole population of bacteria in the Bowel which responsible. Because many of them depends on the whole population, right?
Yeah, so all we're able to say from what's been done so far in IBD is the composition of those bugs in your gut and the interplay with the immune system and immune cells and how they infiltrate and whatever's going on, that they're playing some role, but we don't know the specific bacterium that are maybe the most causal. And you're right, maybe it's a mixture, of those that are causing it, and that's still to be determined. So we basically took these 163 genes, and we had lots of networks that we build in different populations, different tissues for different diseases and so on, and projected these genes over all the networks. And I was looking for networks where the dispersion of these loci in the network was very low, and that they were concentrated in certain subnetworks that may tell me that non-random distribution is indicating that network may be informative for those genes. So here's one of the networks that gave the top hit. This was a network that we built from 800 patients from omental fat, collected from 800 patients. So here, all of the pink nodes and yellow nodes represent different gene expression variables, what we call EQTL, so these are genetic changes that cause changes in the expression levels of genes, or clinical features that are associated with IBD. So all of those nodes, that's what those nodes represent. And then the blue and red edges are the causal relationships that we infer from those variables based on either the genetics. We also bring in as prior information, protein-protein interaction data, protein-DNA binding, protein small molecule interaction, and so on. So we can leverage data that wasn't generated in that population, but it informs on the relationships of genes nevertheless, and we can incorporate that as structure priors in our Bayesian network reconstruction paradigm. So we're integrating, so the bottom line is there's lots of information integrated into this network. And now what you're seeing here in the yellow and red, this is the network that lit up when I projected the 163 genes onto this network. So what you would have expected if this was just a by chance, if the network wasn't really informative, you'd expect a sort of a random scattering of those loci over the entire network. But instead what happened is this particular network, subnetwork, was 5-fold enriched for genes harboring variants that were causing IBD in the human population. The probability you would see this sort of enrichment by chance is on the order of 10 to the minus 100. So it's a pretty significant enrichment, something we're going to pay attention to. And now I can go and say, well, what's going on in that network? So here you're visualizing it as like a lot of data, and you don't know how to make sense of it at that view. But now I can zoom into those yellow nodes and red edges, and I can start getting at some of the structure by now coding, this is where the visualization comes into play of how you can sort of change the kind of information you're being fed. And here what you're seeing is a lot of information from that subnetwork. So these are the yellow nodes. The big nodes here are the key driver nodes. So remember the animation, when I hit that one node and it lit up the whole network, that's what those nodes are doing. So when we perturb those in silico, they have a big impact on the state of the network. These guys are the first, most proximal responders to the key drivers, and then these guys are more downstream, and then these guys are interesting because they're modulating the key drivers. And here you're purple, the edges now are purple if you're activating, and green if you're inhibiting. So now why this structure matters is, remember I said the entire network was 5-fold enriched for genes that harbor variants. Those key driver nodes are 20-fold enriched, right? So that enhancement in the enrichment is telling us that these genes are much more important and we might want to focus attention on them. And then we also look at the information. So now we're visualizing the same network, but now we've coded it in a different way. And what you're looking at here, this pie chart underneath is each of the different colors represent a different biological process. And now the nodes have been ordered or arranged into the different colors that they've been annotated with respect to the biological processes. And now what I'm doing is I'm drawing the edges slightly differently by saying if you're 2 nodes over here and you're communicating with another node over here through a common pathway, then I'm going to collapse your edge down into a little bundle.
Take different category of process or different pathways.
So these are different pathways.
Different pathways.
Yeah.
Maybe gene and protein are the same pathway.
Yeah. And so what's cool that emerges here is you start seeing these main arteries, these sort of the information superhighways of the flow of information in this network. So it's sort of, you think of it as a big transportation system like New York, where you have lots of different streets, and anybody leaving from this part of town, there may be multiple different ways that you can get to another part of town. And so if there are common paths, you collapse those down and you rapidly start seeing the main thoroughfares, the FDR drive and so on. Well, that's what's going on here. You see these main arteries of how one process being affected, that information flows to other pathways and can affect those pathways. So it gives you this much more global view of what's going on. We've applied this exact same sort of thing, not just to IBD, but to many other diseases.
Before you leave IBD, there may be these main pathways, but you can get to your destination many different routes.
Yeah.
So what have we really learned about IBD in terms of the links between these nodes?
So what you then learn is that if you change a given process, and I'll show you an example, if you change a given process, a given set of genes that's in a given pathway, how that information propagates through, whether it's compensated for, whether it shuts too many things down and you defeat the immune system, or whether there's compensatory changes that come and defeat the efficacy you wanted to derive from that. So you can predict all of those sorts of things to come up with where are the best points for therapeutic intervention. And I'll tell a little bit about how we do that screen to come up with new
For one
therapies.
individual, but they may be quite different for another group.
Exactly, and so the whole game then in the genetics and knowing the perturbations that are causing that, at least on the genetic side, is we can understand the constellations and how they're changing of those genetic events, in which processes they're affecting, and then stratify the patient population into those subgroups is the aim. So we've applied the same thing to Alzheimer's, and I only wanted to highlight Alzheimer's because we again, did the same thing of profiling lots of different brain samples, prefrontal cortex and visual cortex cerebellum, and populations, built networks from that, and basically came up with this as one of the key driver networks underlying Alzheimer's, where again we found significant enrichment for genes that are being identified in the GWAS, ones that are being identified in the sequencing study, and from this identified TYRO-BP as a novel key driver of this network, and then validated that guy's role in Alzheimer's disease. Not only that, papers came out while this one was coming out that showed that this new gene TREM2, one of the hardest hitting genes for late onset Alzheimer's disease, identified by DECODE, and I think another group was involved too, but changes in this gene with very high penetrance cause late onset Alzheimer's, and basically this gene signals through TYRO-BP, so we made one of the first connections between those, our new key driver and a new genetic finding, but basically organizing all of this information in the form of a network. And the interesting thing to me about this network is it was identical, nearly identical to the IBD network. So the same thing that was being disrupted in the gut, here to cause IBD, was also being disrupted in the brain to cause Alzheimer's disease. And so we were among the first to make a very solid causal link in the microglial cells of the brain as a key driver of Alzheimer's, not a
We're doing the
consequence.
same thing, same genes. We're
It's
doing
the
like
same, that
thing.
this network is highly conserved between both intestinal, mental, many different tissues, but also the brain. So the connectivity structure of this is highly conserved between those tissues,
When you say connectivity is
including
like,
the same nodes. How the topology of this network is highly conserved.
so the connectivity being highly conserved, is that like an inflammatory pathway?
Yes.
Or it's basically an inflammatory
Yeah.
pathway, so it's all of the it's the gene expression and the metabolites that are involved in inflammation.
Exactly.
But they different genes, yeah?
No,
Are the same genes?
same genes.
The same genes involved.
Yeah.
Are there any evidences of comorbidities between in the same individuals for those?
I mean, there's, yeah, I'm not sure I quite get your question, but there are definitely connections between, in fact, let me first, before I get to you, Gary, let me see, yeah, so let me show you this picture just to give you a handle on how we're looking at it and how diseases are connected, and that some of these are comorbidities that are overlapping. So this is
what kind of genes they are? What kind of my basic function?
so the basic functions are all immune response and inflammation. So sort of the the response of it. So in the Alzheimer's, for example, the hypothesis we're pursuing are yes, you're getting plaques, and of course the plaques were thought to be the driver, but what we're seeing is how the immune system in the brain, the microglial cells, are responding to that is actually what's driving you into the damaged
But you
state.
can miss upstream and by different causes for upstream for most cases, right?
Yes, I agree with that.
Is IBD a risk factor for autism?
What's that?
Oh, sorry.
Alzheimer's?
Alzheimer's.
No.
Yeah, I don't think there's a direct connection, but the way to think about it, it kind of gets to the point this gentleman was making, is that you may have many different types of perturbations that are affecting this network, and the context of how the system responds to that perturbation may be important. So if you have the IBD sort of perturbations that are happening, it turns that network in a way that's context specific to the gut and will cause things to happen, versus a whole different set of perturbations are happening that are more sensitive in the microglial cells in the brain.
It might be helpful if you connected the different, or the similar pathways in different tissues to different downstream pathology. I think that might make it a little bit clearer. Right? Because you're getting very different
Yeah, so and
pathology as a result of.
and that's sort of what this, I think, is going to show. So what we noticed was this particular network that we found in IBD and Alzheimer's, we were seeing in lots of different disease areas. And so what we did was an experiment. This was carried out at Merck in collaboration with Sage after I left. But what we did is we looked at a whole bunch of different diseases. So atherosclerosis, diabetes, inflammatory pain, COPD, asthma, fibrosis, obesity, stroke, and so on. So I think 12 different diseases. And these were animal models for the diseases where we took the tissue that was relevant to the disease, and we looked at the expression of the genes in that tissue in the disease model versus the healthy model. And we looked at what genes are changing between the healthy model and the disease model. And then we are depicting them here in this matrix, where along the x-axis here are the different genes that make up the network I was just showing you, the IBD Alzheimer's network. And you're looking at the expression levels across very varied diseases in very different tissues, aorta, eyelid, skin, lung, and so on. And what you see here is amazing concordance in the expression state of these genes across a very broad set of conditions.
How does this relate to, I'm forgetting the details now, but there was a paper about 2 years ago comparing some inflammatory states in rodents and humans that made some interesting and potentially important observations vis-a-vis the animal models that we use, and I'm wondering how this relates to that. It seemed to have important.
That there may be differences,
Yes,
important
yes.
differences.
Yes.
Yeah, so I think that's likely probably true. What we were focusing on specifically in this study was the commonality, not amplifying the differences. And I think what we're seeing, the fact that the genes that are listed here are human genes from that network and the orthologs, how they're expressed in mouse, and seeing that degree of concordance, I think at least indicates that there's some common degree of response. But I agree with you that there may be very important differences that we could lock onto if we specifically study the differences.
So if I play this back, is this a characterization of an immunoinflammatory response? It doesn't seem like it's as specific per disease. It seems like you're characterizing the whole pathway.
Exactly. I think that's right. And where the context in which that's happening can then predispose to different diseases. If you have that sort of if there's some dysregulation or amplified inflammation response in one part of your body, like in the brain versus the gut, then you're predisposed to Alzheimer's versus IBD. So again, the context of where that's occurring is what's important.
And so out of curiosity, what's the marginal cost to study more diseases? So you characterize 12. If you expand that to 50, is that?
Yeah. So it's more or less a linear increase in the cost. So it's not. I think we stopped here. It was mainly the access to the animals and breeding enough for eat. Like it grows pretty fast because you're looking at different sexes and you're wanting to have 5 to 10 animals per sex, and then just getting the model from people who will share it with you and having it carried out in a common environment. Like if you did 50, it would be harder but doable. Yep.
So is it fair to say that the best drug targets would be to identify that small set of genes that are differently expressed for the different conditions that are associated with driver nodes, and go after those to have a targeted therapy, as opposed to just something like cortisone, which just suppresses
Right.
your whole immune response?
Yeah, so I think that I think intuitively that makes sense, and we do think about it that way. But another way we're screening is with network state as the screening phenotype. So now you have this network, and the state of that network is going to determine whether you're being predisposed to a given disease or not, or being protected from it. So what you're looking at here now are all the blue nodes, or this special inflammation immune network I've been showing. And what we're doing is we're mapping all the different marketed compounds and natural products that are known or predicted to interact with the coding products of this network. So that's what this map is. And the pink nodes are the clinical features of disease that are known or predicted to respond to changes in that network.
But there is no metabolite in here, no?
There is no metabolite data in this particular network, although we do incorporate metabolite data in this. For this network, we didn't have that information.
It seems an important biological question, but even more so for therapeutics, is still explaining why you would get activation in one tissue versus another. Because you're not getting global activation of these systems.
So for example, Alzheimer's disease, you have some hits and you're getting plaque, and that plaque is going to initiate some inflammation response. You don't have the plaque in your gut, right? It's in the neuron. So I think the genetic hits you have that promote plaque development and progression may be one of the things. And then what matters is what's your genetics for responding, for that inflammation response. So I think that's the commonality, but the difference are what are the perturbagens that drove it to that. Does that make sense?
Yes, and those are the things we like to target.
Yeah.
Because that's what's unique to the different diseases, and you limit the perturbation for the entire body if by not going down to the downstream process that.
True, or you could say that the modulating this network has such overall benefit for a range of diseases that you.
Perhaps. Hopefully.
Perhaps, yeah.
And the example you did when you can simultaneously correct a different part of the body in the same network. And the neurons doing that, as you say, and you can control simultaneously and beneficially.
Yeah, so I'll show you how we do that here. So the main thing to take away from this image is that it's pretty freaking complicated. Like lots of drugs and natural products are hitting this network. So the whole idea that you're going to pick this one that hits this one gene and it's going to magically transform everything is probably a little naive. Our computational ability to understand how this multifactorial space is twisting the network, I think, is still limited at this point. So we adopted empirical strategy, which is to, so this was a method pioneered by Joel Dudley at Stanford, who's now in my department, where you're basically looking at what's the effect small molecules have on the expression state of the network, what's the impact a disease has on the state of the network. So you're sort of doing this pattern matching between small molecule and disease. And there's an algorithm that's basically looking at, are you, for this drug, are you pushing those key driver nodes or the network nodes for the disease in the right direction to basically transform that network into a more normal state? And you're doing that empirically, so screening lots of different drugs and just looking at the transcriptional response and then using that as your readout. So that's what was done for IBD with Joel Dudley. where he did this objective screen for IBD over the entire marketed drug space and pulled out, wanted to say what's the top hitting compound that has no indication for IBD, but we're predicting is going to impact IBD, and topiramate was at the top of the list. And so topiramate is an anti-seizure drug, and that drug given to rat. So this is an experiment to validate the prediction that topiramate would have an effect on IBD. There was no physician standing up saying, yes, let's use topiramate for IBD. There was no known association. So Joel carried out the experiment where they chemically induced IBD in a rat model. So here's normal rat intestine, here's chemical induction of IBD, big inflammation, and here's after treatment with topiramate, and here's after treatment with prednisolone, one of the standards of care. And the topiramate in this model looked as good or better than one of the market leading compounds. Again, completely unanticipated connection, but a very data-driven approach to get there. Joel just had another paper in Cancer Discovery where they repurposed a tricyclic antidepressant for small lung cell carcinoma. Again, nobody on the planet was thinking this tricyclic antidepressant would be a treatment for small lung cell carcinoma, but it wiped out small lung cell carcinoma in a number of xenograft models that they looked at for the paper, and is now in a phase 2 trial, clinical trial, given it outperformed in the xenograft models the market leading compound for small lung cell carcinoma. So I know I'm probably getting out of time, but I thought I would say a few words on what we're doing on the psychiatric side, employing the same sort of thing, where when I got interested in the psychiatric space was integrating imaging data, structural MRI, functional MRI, other types of imaging data with molecular data to predict higher order physiological traits. So we basically can take the imaging data here, just clustering the different regions of interest in a MRI study that was done in schizophrenics versus controls, and seeing some of these regions of interest from the structural MRI data, discriminating between cases and controls. But the idea is, how do we link that up to all the molecular information we have now on schizophrenia, given we can't profile the brains of people who are living yet. They don't like to give biopsies of that material. So the people we have the imaging data on are different than the people that we have the molecular data on. So how do we integrate those two together? And
your particular group, because schizophrenia is very diverse, yeah.
Very diverse.
So how you choose the sample for MRI, that will probably be very different.
Yeah, so at this stage, we're collaborating with some of the top centers in the imaging consortia that have come about, funded by the NIH to generate this data, and we're just collaborating with them.
But they take diverse cases, or more or less cases in particular style, because
it's a whole range.
Whole range.
Yeah. So the idea we had was we can integrate the molecular data with the imaging data by leveraging DNA as our common source. So we're storing DNA in both, so if we can look at the genetic perturbations that are driving each, we can link those two together. And so I'll just quickly go over the how we're doing that. So we can take a resource like the Allen Brain Institute Brain Atlas, and what we're doing is we've transformed that data into the MNI coordinate system that's used in the imaging space. So they have pretty high resolution, voxel level data where they've profiled each of the voxels throughout the brain, many thousands. I forget what the resolution is, but it's pretty small, and they did that over a few brains. So what we could do is take the genes that are expressed in a given part of the brain. So here what you're looking at is a particular region of the brain. So each of these nodes now represents different voxels from the Allen Brain Atlas Institute. And they're being correlated with each other if they're sharing genes in common in the given region that they're being identified.
So how do you determine the way which gene is expressed in the given part of the brain in the human?
So they took the human brain and then they cut it up into small little voxels. Postmortem. Yeah, these are all postmortem. Yeah, because that would be hard to achieve. Brain. So this is
sharing gene expression.
What's that?
Expression level.
Say that again?
When you say sharing genes for these different regions, you mean they have the same levels
For
of
for
expression?
this plot, it's just, are you expressed? So
Whether the gene is
it's
expressed.
just sharing, the relationship is the amount of genes you're sharing that are both co-expressed in the given region. So this is a topological, this is a graph from IOSD. It's a topological data mining company. Gunnar Carlson, who pioneered a lot of the topological data analysis, formed this company. And so I won't go into the details of how these get formed, but basically voxel level information being looked at here, and we're looking at genes that are explicitly, more specifically expressed in this part of the brain. So the more red you are, the more localized you are to a given region of the brain. And so you can see this region of the brain has whatever genes are being expressed there are very specific to that. And what we can do is now map on this is a little lower resolution on the imaging data, but we can now voxel per voxel line up the imaging data and the molecular data to see, are the genes being expressed in this particular region also associated with imaging data that's different between cases and controls. So that will tell us what are the genes that are being expressed in the regions of the brain that from the imaging data are different between cases and controls. So here what you're looking at is regions of the brain from the imaging data that are the most different. The red means you're the most different between the schizophrenia patients and the controls, but we can map that now onto the molecular data from the brain atlas to identify, what are the genes that are specifically expressed in the regions that are most different in the imaging data between cases and controls. And once we have that information, so here I'm just highlighting some of the different regions that were the most different between the schizophrenia and controls, and the genes that are specifically transcribed there are depicted in this graph. So now I can take that information and project it onto the molecular networks that we build from brain. And so I'll just jump to that graph. So here's a prefrontal cortex network that was built from both schizophrenia patients and controls. We had around 300 schizophrenic patients, 300 controls. These are all postmortem. So it generated this network, identified regions of the network that were most involved in schizophrenia. So this is basically projecting the genes that are different between cases and controls onto the network, and they localize into these two networks. But now I can associate the region that's changing in the imaging data to this subnetwork through the Allen Brain Atlas, because I know what genes are being specifically transcribed in that region. So I can take what's being specifically transcribed in this region, project it onto the network, and the network it hit was this guy right here. So pretty cool. Like, we wouldn't have been able to make that connection because we had no way to bring those two coordinate systems together. We didn't have matched samples, but using the Allen Atlas as the connector, we were able to make this link. Here are the pathways that are involved, which when I show up to the schizophrenia people, they're excited because they say those are genes that are involved. And now I can pop in to the network, and so this is just a portion of the network that's involved. And now what you're looking at are the greens are the genes that identified in the schizophrenia GWAS, so variations in these genes cause schizophrenia. The blue nodes are the key drivers of this subnetwork. And so now we're already locked onto things like GPR37, which is connected to multiple genes from the human GWAS for schizophrenia. And so now pursuing that as a target for schizophrenia, again, a novel finding in addition to other key drivers. But that's the sort of information that in the psychiatric side is pretty transformational to those guys, because they before that had no way to hook up all that high dimensional imaging data to the molecular information. That's what we're doing. Yeah.
Excuse me, I didn't quite understand how you went from the novel data set of the schizophrenic and control human brains and how you merged that with the Allen Brain Atlas. I didn't quite get the mapping.
Okay, so here's a network that we built from prefrontal cortex, about 600 individuals, half have schizophrenia, half don't. But we don't have imaging data on these people, right? We just have, their brains, post-mortem. So that's what this network is. Then this network is from imaging data, a whole nother population of like 1,000 individuals that all had MRI. So this is structural MRI data. So this network is showing us the structural MRI data in the regions in the MRI data that are most different between cases and controls. So in the imaging data, this part of the brain has the biggest differences between those with schizophrenia and those who don't. And so what we did in the Allen atlas is we said, well, what genes are specifically transcribed in that part of the brain? Like that's what the Allen atlas gives us, right? And then we take that and we project it onto the network. And in that way we can connect the schizophrenia part of the network that's associated, the part of the network associated with schizophrenia to the part of the brain that the imaging data tells us is associated with schizophrenia. Does that make sense?
Yeah, that makes perfect sense. And then what is the original data? What's the data you're taking from the brains, the dead brains? Is that slices? I guess I understand everything now except for what created the data.
So what created this data was, so I don't exactly know how so it's prefrontal cortex, and I forget the exact region, but